this video, we will go through the first module of LNCRNA-KB, which is the Browse Genes module. In this module, users can actually search for any of the genes that are part of the long non-coding RNA knowledge base. There are several ways to search for a gene. There is a quick search option in which you can use any of these IDs, for example, the gene name or gene symbol. You can use the ensemble ID, you can use the long non-coding RNA knowledge base ID or LNCKB ID. You can also use any of the IDs of the 6 annotations that have been incorporated into LNCRNA-KB, for example, the Phantom ID or the Chess ID. You can use the Linkipedia ID, non-code ID. In addition, we give the user a advanced search option by clicking on this search box in the previous screen that you saw. You can actually use any of these fields to search for genes or to narrow down your search. For example, your coordinates of the gene, the type of gene. For example, you can say long non-coding RNA or LNCRNA, and this will give you just the LNCRNAs that are part of the knowledge base. A cool feature of this is you can export the list as a CSV file or Excel file to further interrogate or further use the list for other purposes. You can also search for other types of genes, for example, protein coding genes, which are part of the LNCRNA-KB, and this will give you just the list of protein coding genes in the database, as you see here. And in the bottom, we actually give you the numbers that your search produced, the number of genes. But for actually walking through one of an example of a gene, I would like to go through this search option and show what details of a individual gene that we give or provide on the LNCRNA-KB. For example, I will use the example that I gave in the manuscript, a commonly studied long non-coding RNA MALAT1, which I provide in the technical validation section. And using that example, we will go through all the details available for a particular gene on the long non-coding RNA knowledge base. So going back to our table of all genes, as you can see, these are all the genes that are available to browse, for which we have information on or that have been incorporated into the long non-coding RNA knowledge base. As a quick search option, I will just search for MALAT1, and you hit the enter key, and as you can see here, we get the criteria that we were, the gene that we were searching for. So you can see in the description, this is the MALAT1 long non-coding RNA, all the information, some of the information. And then when you click on the LNCKB ID, gene ID, it takes you to the gene page or the gene info. And within the gene info page, right off the bat, you can see that there's several information available for this gene. First and foremost, we have the gene info itself. So by clicking on any of these tabs, it will actually jump to the section for that particular criteria. For example, we have the tissue-specific expression of MALAT1 or the tissue specificity scores that we calculated, whether this the tau and the PAM scores. We also have the network and pathway information for that gene and the EQTL information for MALAT1. As you can see, you can get all the information for that gene, the IDs, which source it was incorporated incorporated from. So MALAT1 was incorporated mainly from Chess, and by clicking on any of these IDs it will take you to the actual database or the actual annotation. So in this case, it will take you to the chess annotation, and if it's a searchable annotation database, then it will take you to the gene page of that specific gene in that annotation. Also, we have links to external links to the gene cards. We have external links to Ensembl and NCBI gene, and so forth and so on. For the tissue expression, as you can see here, we give the expression across all the tissues, and this is a dynamic plot that we generate for each gene. There are several functionalities available. You can zoom into this plot. You can save this plot for use in your manuscript or in your presentation to show the expression of this long non-coding RNA across the 31 tissues that we analyzed in the GTEx using the GTEx data. We also calculated the tissue specificity scores. Here we give the tau score, which actually gives the idea whether this gene is specific to is tissue specific. The tau score ranges from 0 to 1, as well as you have the PAM score, which actually tells you for each individual tissue whether a particular gene is specific to, and this is the distribution of PAM score across the GTEx data that we analyzed. Here we have the tissue expression network module information. It gives you the top 3 pathways across all tissues for which the MALAT1 is involved in. Basically, it's the co-expression network analysis of gene expression data for the 30 tissues to which MALAT1 is assigned to a particular module or cluster of genes. And within this, you can actually go to the specific network and enrichments page by clicking on any tissue. So if you click on brain, for example, it will take you to the network and enrichment pages for brain tissue, and you can actually get all the information, which we will explain in a separate video. You can also download all the information, all these tables as a CSV file. It also gives you all the Go terms in which MALAT1, the top 3 Go terms for a particular module in which MALAT1 is involved, the number of link RNAs in that module, and you can also download the module adjacency matrix right from here in which MALAT1 is part of. We also give the EQTL information for all the EQTLs in MALAT1, which have a raw p-value of less than 0.05 across all the tissues that we've analyzed in GTEx data, a bar chart, as well as the statistic, the p-values for particular variants or SNPs in which the MALAT1 has an EQTL of p less than 0.05. By clicking on any of these, it will take you to the actual DB-SNP, externally linked to the DB-SNP page, in which you can actually view all the information about the variant. So you can also download the entire table across all the tissues, and other parts of information available in the gene. For the gene is the transcript information, in which we give all the transcript details for a particular gene. Here we show all the transcripts that are part of MALAT1. We give a table, all the coordinates and the positional classification of these transcripts. For example, where a particular transcript is in relation or with respect to the mRNA or to other mRNAs. For example, this transcript shows that it's basically genic, containing this gene, and it's basically also we calculated a coding potential score, which tells you whether a particular transcript is potentially or likely to be coding. So in this case, based on certain criteria and classification algorithm that we use, it tells you whether a particular gene or transcript is potentially coding or not, and that is denoted by a 1 or a 0. Also, we give you the dynamic transcript expression plot. Basically, by clicking on any transcript, you can get the expression of that transcript across the tissues that we've analyzed in the GTEx data. And in addition to that, we also calculated the conservation score across all the exons of a particular gene, and in this case we merged all the exons together across all the exons and overlapping exons, and you can see the mean conservation score using the 30-way conservation track. And here are the different statistics or metrics for the conservation score, which are all downloadable and searchable in the transcript details page. In addition to the transcript details, we also give you the trait association for that particular gene, for in this case MALAT1, in which MALAT1 has a association with a particular trait using the summary Mendelian randomization algorithm for this across the all the tissues, and we've tested approximately 323 GWAS traits from the UK Biobank. And this table basically shows you all the traits in which MALAT1 would have a significant p less than 0.05, summary Mendelian randomization p less than 0.05 across different tissues. And using this table, you can see if there's any particular gene that was prioritized, whether this gene, sorry, was prioritized across different traits in different tissues right off the bat. We also have the genome browser that we've incorporated, and the genome browser basically shows the different transcripts variants that we've aggregated across, that we've actually accumulated across the 6 different databases. We show a track of all the transcripts in addition to other tracks that can be turned on and off through the UCSC genome browser, as shown here. And this is the custom track coming from the long non-coding RNA knowledge base. So this is all the information that is available for a particular gene, and it's all downloadable, it's all viewable using the via the Browse Gene page.